So this will take us to our next speaker. So I'm happy to introduce to you Dr. Roland Ewald. Roland completed his PhD at the Computer Science Institute at the University of Rostock, where he also worked as a research scientist. 2014 to 15, he was taking the lead of a software development team at a clinical laboratory for genetic diagnostics. This is also where he learned about the challenges and opportunities with clinical NGS diagnostics. And together with colleagues, he founded Limbus Medical Technologies, and he's now the head of the industrial engineering and IT team. So he's currently leading the bioinformatics and cloud infrastructure team. Thank you, Roland. Welcome.
Thanks for the introduction, Yvonne. And yeah, also from me, welcome everybody. It's great to see so many people interested in this. And I realize this is your lunch break, and this is kind of a technical topic, because transcript alignment is really something that's very bioinformatics specific. But to just make it clear what the importance is here, there are imperfections in the transcript alignments, and we call them mapping gaps. And these mapping gaps can actually lead to invalid variant annotations. And I'm not talking about like some niche annotations here. I'm talking about things that are really clinically relevant, like cDNA annotation, protein change annotations, the things you look at every day, and where you basically very quickly want to see, does this variant have a certain impact, you know? So to be clear, and I don't want to be alarmist here, so this is only a very small number of transcripts. So we're talking really like the vast majority of transcripts is fine, but there are a few transcripts that just can't be aligned perfectly to the reference genome. That's just a matter of fact. And in this talk, we are going to focus on RefSeq transcripts, because from our point of view, this is like the clinical standard. Of course, let's say, just the fact that there are some transcript alignment problems doesn't mean really much, right? So in my talk, I want to basically answer 3 questions for you. The first one is, does it really matter? So is it actually even clinically relevant right now? Of course, we just heard from Rami that things that are not clinically relevant now may very well be relevant in the future, but still would be nice to kind of get a feeling for that. Then later on, I will dive a little bit more deeper into the bioinformatics of that issue and just show you how you can analyze these kind of cases. And finally, I will try to give you some recommendations of what we found works in practice, how to deal with that. So yeah, what is the impact? The first question is, of course, people would maybe ask as well, I'm using the new reference genome, right, HC38, so maybe I'm not even affected, right? So what we did is we analyzed if this problem went away, this HC38. And it got better, but it didn't go away fully, unfortunately. So here are just some statistics. For the latest RefSeq alignments, we see that 1.4% of the HC19 aligned transcripts have a problem, and for HC38, that's still 0.2%. But still, that's like 54 genes that have transcripts with mapping gaps. And well, that still doesn't tell you so much, because these are just genes. Are these clinically relevant? And to answer that question, we thought of, let's say, how to query our databases. And we came up with some criteria. And to keep it very simple, what we tried to, what we decided is that we consider a transcript clinically relevant when we find it to be coding. So in RefSeq nomenclature, it means that the accession of the transcript starts with 2 letters N and M. So it's a coding transcript, it codes a protein. And we also require that the gene of the transcript has at least one association with some phenotype in HBO, right? And that narrows down the list of these problematic genes considerably. We're now talking about 70 genes for HC19 and only 13 genes for HC38. But so for your convenience, these are the genes that have transcripts that are problematic. But then we went, of course, to our reference database, right? So our customers share data anonymously to improve their diagnostics. And of course, we can now find out from this database, we process thousands of samples each month. Let's say, are these transcripts even used? And for HC19, about 1.8% of the clinical reports and allexes, that's currently about 25,000, so we have like 70,000 patient records in there for about 2,500 diseases, different diseases, and 1.8% of the allexes reports for HC19 do refer to at least one of those transcripts, so to say. And yeah, so that's quite a bit, but of course that's not the whole story, because The next interesting question would be, well, is it evenly distributed? Now, do I have to care for all of that evenly, so to say? And we found that no, that's not the case. Basically, these kind of cases are currently focused on only a handful of transcripts. So if you are dealing with those, you should really be very careful, so to say. Now, of course, let's say for HG38, you see basically 80% of all the cases would just be, it's just about 2 genes, right? It's about PTEN and ALMS1. And then, of course, SHANK3 is also another, let's say, kind of well-known gene that has these issues. To kind of illustrate for you why that is, why these transcripts are being used by clinicians. I'm sure most of you know that, but let's say for me as a bioinformatician, it's always interesting to check back. I just want to show you what the RefSec team kind of recommends. They have this kind of expert group called MAIN that recommends transcripts, and of course you can now basically check for each of these genes. Well, maybe I have better options, right? Maybe I don't have to use these kind of challenging transcripts. So there's this, for example, for PTEN1, there's a MAIN recommendation, and the transcript with the alignment gaps is also listed, of course, on the website. And you see, well, you know, the transcript with the alignment gaps, it has a lot more protein sequence. So it makes actually clinical sense to look at it. The same is happening for ALMS, right? So there's a MAIN recommendation, and the transcript with the alignment challenges is a little bit longer. There's like one amino acid more, and the transcript sequence is also longer. And ALMS1 is actually even more tricky because you know there are different expert groups trying to curate also transcript recommendations. And so in this case, the main expert group said, let's use this transcript, and the LRG group, like the locus reference group, they are recommending the other transcript. So what are you going to do, right? So it's a kind of a conundrum. And to kind of delve a bit deeper into that, I'd like to just show you how you can analyze these kind of issues for yourself and how to deal with that. So what is actually the cause of the problem? The cause of the problem is just fundamentally that some sequences simply, also for biological reasons, don't really match the reference genome and probably never will be. So, and the RefSec team recognizes that, and they still try to provide for for potentially clinically relevant transcripts alignments. And that is basically this orange arrow here. That is where the trouble starts. So if the DNA sequence and the RNA sequence simply can't be brought together, so to say, then we always will have these so-called mapping gaps. And these have to be considered when you annotate the variant, because where you think it is, when you think of the genome, is not where it really is on the transcript. It's just a different coordinate, so to say. And that is where you have to be really, really careful. As we will see, what is also important here is that these transcripts, alignment algorithms, they are getting improved, right? And these are like large pipelines, bioinformatics pipelines. And of course, there are new transcripts being discovered. So RefSec regularly updates their alignments, right? And this can also have an impact. So basically, what you need to take into account always is that the transcript alignment is not fixed and it's not perfect. Here's just, let's say, my favorite software to check alignment issues. That's the NCBI Genome Workbench. They have a great YouTube channel, so just check it out. It's actually fairly easy to use, and they have some nice tutorials for that. And here's just one example. MUC2 also has a transcript that is challenging or cannot be mapped completely to HG38. And what you see on top is a reference genome, and on the bottom you see the transcript, and in the middle, these two gray stripes, these actually show you the alignments. So if you look at this green box, this is basically the symbols. These are the symbols for a match. So this is like a good reference. And then you have also so-called basically SNPs, but SNPs between the transcripts and the reference genome now. And so mismatches. And of course, you can also have insertions and deletions. Very similar to what you would find in a case, right? But you have the same principle here as well. And these kind of imperfections need to be taken into account. And as I said, these transcript alignments can change. So you see there are two gray bars here, right? So you need to take into account which alignment you actually use. The upper alignment has one insertion here. The lower alignment has two insertions here. So what I'm trying to get at is that, let's say, what we are used for medical practice by basically just saying, well, that's a gene and there's a protein change to describe a variant like that will not be sufficient. And of course, let's say this is common knowledge. Bioinformaticians will tell you, well, use a transcript and actually use a transcript with a versioned accession so that the transcript sequence is actually fixed. And my point is that's not really enough to even define, coming from the genomic coordinates, what the cDNA is, because you also need to take into account the alignment version, which can change. And well, that's why we actually would always need also to fix the alignment version here and to document that. And well, let's say that is not the ideal solution, right? So I know it's a lot and it's already tricky enough. So what are we doing now? And there are multiple things you can do, and I just want to show you a few, give you a few recommendations here. So the first is that, let's say, the software and the bioinformatics team at varvis are really aware of that issue, and they are working quite a bit on that. So we have a long history of, let's say, improving corner case by corner case, this kind of annotations. And there are literally millions of corner cases here because of this kind of combinatorial explosion. There are forward and reverse strand, different kinds of variants. You have splicing and so on, and all these kind of additional analysis need to take into account these gaps. What we also offer, apart from our own annotations, are of course link outs to other databases. So what you should be doing is, of course, if you are in doubt or if you are working with one of those tricky transcripts, you should check the other databases, right? For example, dbSNP. This is just one example from ALMS1, one of those transcripts that is one of those genes that has a challenging transcript. You can check ClinVar as well. And whenever, let's say you use your own in-house bioinformatics tool or like some other tool, whenever you find for the same variant a different cDNA, let's say my gut feeling would be 95% chance this is because of a transcript. It's not a normal bug, it's just an alignment issue. It's a mapping gap, so to say, right? And that is important. And actually in this case, you see maybe this is just 3 base pairs difference. So it's just a next amino acid. It's just three bases difference. And that would be where you can now use your new knowledge about the NCBI genome workbench, open the NCBI genome workbench at this position, and you find that briefly upstream, there's actually the mapping gap. So there are 3 bases missing, and that's the 3 bases that have to be taken into account. Otherwise, the cDNA annotation and the protein change, and also potentially the sequence ontology annotations or putative impact would be wrong. Another thing that you should be doing, also in terms of regulations, is you should be documenting the alignment that you're using. So we had this like a while, but it was not really, let's say, present enough. So in the newer varvis versions, we now present this really as an annotation source. So you will update this like you will update any annotation source, like you run a ClinVar update. It's just basically an input for your variant annotation pipeline, so to say. And this is also something that will be shown on the reports, on the clinical reports you can generate with varvis, because it's just important context information. And the next recommendation I would have is go back there from time to time to these kind of tricky genes and reconsider the expert opinion, so to say. For example, the main group, I think, has now basically succeeded LRG, so at least considering their website. Maybe their recommendations are more recent, and maybe there's a good reason why you don't have to use these kind of challenging transcripts, because it kind of reduces the probability for error, also with your own tools, maybe, if you don't have to use them, right? So I'm just saying, reconsider the transcripts you are using and use these kind of challenging transcripts for good reason. And well, so this is just a view and how to do that in varvis. I'm sure you can do that in other software in a similar way, right? It's just, my point is, you should be checking that. And let's say in varvis, what we also show you when you try to select a transcript is, we try to give you more information, because this is actually really, I think, a communication issue. You need to really be aware of these kind of mapping gaps. And the RevSec team is actually very helpful, and they are very thorough. So they will give you in these annotation files that the bioinformaticians take to prepare alignments, they will give you comments on the alignment quality. And we try to provide you the same comments so these are from the RevSec curators in the software, so that when you make a selection, what is the correct transcript to use here, you have, let's say, the full context. Let's say, is it a good alignment or not? And so that's what you basically see here. And The other thing that needs to be communicated is, of course, as we already heard a few times, you have to communicate it in your team, right? And for that, we also have a new feature that is basically, it's called a gene comment, basically. So you can just put in some, maybe a hint or a link to an SOP or something like that, so that your team knows, oh, wait, during diagnosis, oh, that's one of those challenging genes. I should check this again, you know, the annotation, if that's actually correct. Yeah, so that would be my tips. So just to summarize, okay, so these mapping gaps, what do they impact? Are they really important? And yeah, unfortunately they are, because they can lead to wrong variant annotations and thus also kind of wrong diagnostics. And well, the fundamental issue here is really different sequences. And so this fundamental issue will not go away, not even if we have a new reference genome, because as I said, there are also some biological reasons why these sequences may not really match as well. And yeah, what can we do? So I hope, let's say we already raised awareness a little bit, because you all listened to this talk. I think the other two pillars in the lab is really documentation and communication, and what we are trying to do to get better, because this is something, just like Rami mentioned, over time you need to get better, you need to learn from your mistakes. Is what we are doing is we collect challenging variants and try to, well, we run automated tests, right? So we try to really collect the tricky cases and the corner cases and make the software better continuously. So if any one of you has like a really challenging variant, we're happy to talk at the booth, try it out and put that into our test set, so to say, right? And we're happy to collaborate on that, because I think it's an important issue to really benchmark all the software against these kind of challenging variants, so that you don't overlook anything. Yeah, so thanks a lot for your attention, and yeah, if you have any questions, I'm happy to answer.
Any questions? Okay, actually, we have one from the audience, the virtual space.
That's where the bioinformaticians
is from Edgar Verdura. Can you provide any insights on main use? Can using them solve any of the problems that you're describing?
Sorry, can you repeat the?
Can you provide any insights on the main use, Mane transcripts? Main transcripts?
Well, I think, let's say, so from what I understand, they have some kind of algorithm, so to say. They have a rule set, how they select transcripts. And I think, let's say, when you look at it, it makes sense. I can't really comment, let's say, on let's say, the epistemological value of their recommendations. So it's like, is it a super strict guidance that you should never to use another transcript or not? So I can't really give you so much detail there. We would always, let's say, recommend customers to use these kind of main select transcripts first, so that you only really override the recommendation when you really know what you're doing. Also, you have to keep in mind that, at least in varvis, you see all the transcripts, right? So it's not that you are then blind. You know, the transcript selection is something that's just, let's say, a default. Right? So even for other, even if you have other default transcript, you will still see the tricky transcript, so to say, in varvis, and you might still be wondering, that doesn't look right here. Why is it, for example, showing C is going to C, you know, or G to G? Well, that's because one is genomic, and the transcript is actually not even a variant. You know, it's actually it's a wild type, so to say. So. Yeah, apart from that, I can't really, let's say, comment on the way the Mane expert group is curating the data set. No, I'm sorry.
Thank you, Roland. If ClinVar is using transcript A and they have a mutation at point 1, and I'm using transcript B, and I have mutation on position 2, but actually it's the same of 1 if I use the other transcript. Is it going to be automatically called as being described in ClinVar, or you would say you are using the wrong manuscript, the wrong transcript, so it's not going to be compared to ClinVar?
Yeah, that's a very good question. So, like internally, because of exactly these kind of challenges, and also HGBS and nomenclatures, the details are incredibly complex, right? So it's really something you don't really want to rely on computationally. Internally, varvis uses genomic coordinates only, because also the ClinVar imports that we do on a monthly basis, they also provide the genomic coordinates. You really need to match against the genomic coordinates for hits. But of course, then since we link out to ClinVar, you're absolutely right. You would have to check, does this transcript make sense, right? Is there additional detail information on ClinVar that prescribes, oh, we are talking about this transcript in particular, and so. But the matching will work because you see these transcripts at such an additional layer of complexity that it's not a good idea, also engineering-wise, to do the matching any differently. So we really only rely on genomic coordinates for matching, and not on transcript coordinates. Also because it changes. And this is the same, for example, for these ClinVar alerts that you get, right? It's all genomic coordinates. Of course, we can always think of ways to make this better, maybe additional hints if there's other transcripts to be used, or maybe like an automated, let's say, suggestion. Look, there's a new transcript, maybe you want to change the default transcript. We have lots of ideas in those directions, but let's say the principal matching is always genomic, because that's the coordinates that don't change. Yeah, thanks.
Sure. Do you have any suggestion to curate transcripts that for genes that have a broad spectrum of phenotypes? For example, PAX6, not all patients have the same type of phenotypes, and then there are transcripts which are not clinically relevant, but then there are some variants there, and then do you have any suggestion to curate the transcripts that could be clinically relevant for subtypes of phenotypes with a broad disease?
That's a really good question. So let's say, as I said, so in varvis you see all the transcripts, and let's say you would, we do have different, let's say, annotations for that. So because we realize that this kind of approach with the full transcript works for many cases, but not for all cases, right? So what we do is we have one column that basically always gives you the maximum impact over all transcripts, so that you are not, let's say, that you're not overlooking a variant where you say, well, this is synonymous on my default transcript, but actually that's a stop gain in another transcript, and you really want to see that, right? So for this we have actually extra columns. You saw these grids, right? So you can filter against, really, I want to see, for these kind of cases, I want to see over all the transcripts in the gene, I want to see the maximum impact, and I want to see what the maximum impacting change would be, so to say. Apart from that, I'm not even sure if, let's say, it's a good idea to manually curate that, right? So let's say our approach is more to try to give the users better features so that they don't have to, because manual curation is always fraught with error, and you need to document this carefully, and yeah, it's tough.
Kind of demonstration about.
Oh yeah, you can just drop by our booth, and we have videos and everything. Sure, it's fine. Sure.
Okay, thank you again, Roland.